Welcome to the fascinating world of cellular respiration, where we explore the intricate processes that occur within our cells to produce energy. In this video, we will delve into the fundamental concepts of cellular respiration, its importance in sustaining life, and the various stages involved in this process. Firstly, let's understand what cellular respiration is. Simply put, it is the process by which cells convert the energy stored in food molecules into a form of energy that can be used by the cell. This energy is in the form of ATP, or adenosine triphosphate, which is the primary source of energy for all cellular activities. Cellular respiration is a vital process that occurs in all living organisms, from the tiniest bacteria to the largest mammals. Without cellular respiration, life as we know it would not exist. It is the process that enables us to breathe, move, and carry out all the functions necessary for survival. Now that we have a basic understanding of what cellular respiration is, let's take a closer look at the stages involved in this process. In the next segment, we will explore the first stage of cellular respiration, glycolysis. Glycolysis is the first stage of cellular respiration and occurs in the cytoplasm of the cell. It is a process that breaks down glucose, a simple sugar, into two molecules of pyruvate. This process also produces a small amount of ATP in NAD, a molecule that carries electrons. The first step of glycolysis is the investment phase, where two ATP molecules are used to activate glucose. This results in the formation of fructose-16 bisphosphate, which is then broken down into two 3-carbon molecules of glyceraldehyde-3-phosphate. In the second phase, the pair phase, the glyceraldehyde-3-phosphate molecules are converted into pyruvate. This process generates 4 ATP molecules and 2 NAD molecules. Overall, glycolysis produces a net gain of 2 ATP molecules and 2 NAD molecules. While this may seem like a small amount of energy, it is an essential step in the process of cellular respiration. The pyruvate molecules produced in glycolysis are then transported into the mitochondria, where they are further broken down in the next stages of cellular respiration. In the next segment, we will explore the second stage of cellular respiration, the citric acid cycle. The citric acid cycle, also known as the Krebs cycle, is the second stage of cellular respiration. It occurs in the mitochondria of the cell and is a series of chemical reactions that further break down the pyruvate molecules produced in glycolysis. The first step of the citric acid cycle is the conversion of pyruvate into acetyl-CoA. This process generates NAD and carbon dioxide as byproducts. The acetyl-CoA then enters the citric acid cycle, where it combines with oxaloacetate to form citrate. Through a series of chemical reactions, citrate is broken down into a 4-carbon molecule, which then undergoes further reactions to regenerate oxaloacetate. During this process, NAD, fat H2O, and ATP are produced. Overall, the citric acid cycle produces 2 ATP molecules, 8 NAD molecules, and 2 FAD-H2O molecules. These molecules carry high-energy electrons that will be used in the next stage of cellular respiration, the electron transport chain. In the next segment, we will explore the electron transport chain and oxidative phosphorylation, the final stages of cellular respiration. The electron transport chain and oxidative phosphorylation are the final stages of cellular respiration. They occur in the inner membrane of the mitochondria and are responsible for producing the majority of ATP molecules. The electron transport chain is a series of protein complexes and electron carriers that transfer high-energy electrons from NAD and FAD-H2O to oxygen. As the electrons move through the chain, they release energy that is used to pump protons across the inner membrane of the mitochondria, creating a proton gradient. This proton gradient is then used to power ATP synthase, an enzyme that produces ATP. As protons flow back across the inner membrane through ATP synthase, the enzyme uses the energy to combine ADP and inorganic phosphate to form ATP. Overall, the electron transport chain and oxidative phosphorylation produce a large amount of ATP, with each NAD molecule producing 3 ATP molecules and each FAD-H2O molecule producing 2 ATP molecules. In addition to producing ATP, the electron transport chain also plays a crucial role in removing oxygen from the body. Oxygen is the final electron acceptor in the chain, and its reduction to water is essential for maintaining the balance of oxygen in the body. In the final segment, we will summarize the key points of cellular respiration and its importance in sustaining life. In summary, cellular respiration is the process by which cells convert the energy stored in food molecules into a form of energy that can be used by the cell. It is a vital process that occurs in all living organisms and is essential for sustaining life. Cellular respiration occurs in 3 main stages: glycolysis, the citric acid cycle, and the electron transport chain and oxidative phosphorylation. Glycolysis occurs in the cytoplasm of the cell and breaks down glucose into pyruvate, producing a small amount of ATP and NAD. The citric acid cycle occurs in the mitochondria and further breaks down pyruvate, producing NAD, fat H2O, and up. The electron transport chain and oxidative phosphorylation occur in the inner membrane of the mitochondria and produce the majority of ATP. Overall, cellular respiration produces a net gain of 36-38 ATP molecules per glucose molecule. This energy is used by the cell to carry out all the functions necessary for survival. In conclusion, cellular respiration is a complex and fascinating process that is essential for sustaining life. By understanding the fundamental concepts of cellular respiration, we can appreciate the intricate processes that occur within our cells to produce the energy of life.